Protecting California's Chinook salmon is a challenging task. There are 4 different run types of Chinook, 2 of which are listed as endangered or threatened under the Endangered Species Act. DWR has been tasked with creating a juvenile production estimate for the threatened spring run Chinook to better understand the size of their population and inform incidental take limits for the run. The problem is that all 4 run types look exactly the same, so we can't tell them apart by looking at them. Because we can't visually distinguish between the run types, DWR has developed a genetic monitoring program which will allow us to determine run type using just a swab of mucus from the fish.
The specific technology we'll be using for this project is called Sherlock. It was originally developed for viral diagnostics, but has very recently been applied to conservation biology.
This technique leverages something called CRISPR, which stands for Clustered Regularly Interspersed Short Palindromic Repeats. And actually it was discovered because it's an immune system of bacteria, more or less. So it's a way that bacteria can recognize the DNA and the genetic material of foreign invaders like viruses. And we can leverage that property for really specific detection of DNA from just about anything we want. Spring run salmon has different DNA than winter run salmon or fall run salmon. So using those specific detection properties of this CRISPR enzyme, we can actually engineer it to detect the DNA from spring run, winter run, fall run salmon. We can take a swab from the surface of a fish, take some mucus, which has some DNA incidentally in it, put it in a tube, put that in another tube, let it sit, and if that specific DNA for a specific run type is detected, it'll glow, it'll fluoresce.
Sherlock has an advantage over some previous genetic tests because it's extremely rapid. You can swab a fish and get the run type results within 30 minutes to an hour. So you could use this in the field. We also have a full genetics laboratory here, so we usually will collect the samples from the field crews so that then we can process all the samples at once because we're getting thousands of samples at a time.
Chinook salmon in California are found in a really wide geographic distribution, and so what that means is we need to be able to collect the samples as efficiently and effectively as possible. We are leveraging our partners across different state and federal agencies to make that happen so that we can get the samples that we need to do the genetic analyses as quickly as possible.
Ultimately, the goal of this project long-term is to help salmon populations recover. And these genetic data are going to help managers in the field make real-time management decisions that will get these salmon back on the path to recovery.